Hi, my name is Keenan Hardert, and I'm a first-year faculty in the biology department here at Gustavus. Today, I wanted to kind of go over some of my research at the Mayo Clinic, which mainly covers lymphomas and how we can harness genetics to basically perfect the treatment for each individual patient. So for this project, I worked really closely with these two clinicians, Tom and Greg. They are just excellent mentors when it comes to, kind of seeing the clinical side of everything. And then my scientist mentor, Ann Novak, is excellent. So if anybody is looking to grad school or anything like that, I would be happy to put you in contact with her. Working with a clinical and scientific team has made what I do very exciting because it's very translatable immediately to patients, and we can harness genomics now in this day and age that can translate right here, right now to every patient. That wasn't possible 10 years ago when sequencing, the exonic regions of, all these genes would have cost, let's say, $5,000 to $10,000 to $100,000 per patient. Today, we can sequence what we need to make clinical decisions about what therapy is going to work best for each cancer patient for maybe $500. Now, can't promise price on everything, so don't go asking your doctor for sequencing. But in any case, we are at an age where we can personalize medicine with genomics. So kind of as some guiding questions for this presentation, which is a new faculty presentation, but I just can't do it next year since I'm only on a one-term contract, but I still wanted to. Can we predict the future of a patient by using genomics? Is this something that we can do? Is this something that we can be sure of? And in this case, can we personalize this? So one of the biggest things with cancer and what I study is that every person has their own genome and their own DNA. Now, when a tumor starts getting aggressive, and I'll let you look at this figure right here, this DNA starts changing. And the driving forces of this pathway, they are going to splinter. There's going to be different DNA changes in this tumor than there are in the normal cells, for example. By using this and harnessing it and saying, okay, based on the damaged DNA sequence that we see in this tumor, we're going to treat it with, X, or we're going to treat it with Y instead. That's pretty bad Y, but look, this is the future of online learning for Gustavus. Sorry. Come on. There we go. It's a Y now. So we can decide treatment based on what sequence of DNA, basically mutations or mistakes, are driving this cancer cell division. Okay, so with cancer, again, this is a good chance to kind of stop and, for everybody in the audience that wants, that is kind of newer to biology or it's been a little while. Cancer is the unregulated multiplication of cells. You start with one cell, and cells usually divide into two cells, as you see right here, for example. Those two can become two more, and it's going to grow exponentially into the point where it is uncontrollable. In a normal cell, you are only going to see a few divisions. There are a lot of checkpoints that stop this uncontrolled type of growth in a normal cell. If you lose those checkpoints, which are DNA genes, you can lead to something like this. So essentially, to get to a tumor, which is what we see here and what patients present with, you need to have almost unlimited capacity for growth, multiplication, proliferation, and another key thing that we won't strictly cover in this is that these cells need to survive. The cells in your body usually have a half-life. They have a timer on them for how long they're going to survive. You renew new cells by dividing more, and they replace those old cells. But if you have cells that are growing and they can survive, that's where you start having a problem. Okay, so this slide just kind of goes over what cells, kind of comprise everything. And they are the building block for your tissues, which become, the building blocks for your organs, the systems, and the body. To keep your body going, you need to be renewing your cells all the time. Every minute, every day, all the time. You are made of trillions of different cells. Now, each of these cells occupies a different part of the body. So right now it's showing the musculoskeletal system. You have neuron cells, you have lymphatic cells, you have intestinal cells, you have all kinds of different ones, and they have to renew all the time. So to renew, they can't just come from anywhere. The cell cycle is something that's very controlled and it's very, guarded. What we need is to take one cell and make it into two. And like we saw up in this slide, that needs to happen, billions of times a day. this is a very important process to renew the body and make sure physiologically you're doing fine. So this is the cycle in which one cell is going to become two. This specifically, this little mitosis section, this is where the cells actually split. That's the actual split right there. And one cell becomes two. So as you're going along this cycle, there are lots of checkpoints. There are proteins in your cell, so little components that say, okay, are we ready to cross this checkpoint right here? Are we ready to cross this checkpoint right here? Are we ready to cross this checkpoint and actually divide? Okay, so in your normal cells, this whole process is tightly controlled. There are tons of stop and go signals. Okay, there are lots of go signals in your body that are going to provide this go, but there are lots of stops at these checkpoints that are like, okay, are we actually ready for this? That balance is the key. When you have an imbalance in those stop and those go signals, this process that turns one cell into two can basically go haywire. And one cell is going to turn into hundreds of thousands of cells instead. Okay? So how do we get to that point, though? In a lot of cases, it is genomics. So everybody has a DNA genome. Every single cell comes complete with all 23 pairs of your chromosomes. The whole thing is in every single cell. Now, in these blueprints, in this chemical nucleic acid, these are instructions for making a transient molecule called RNA. All this is basically a copy of this DNA nucleic acid. It's a little different. It's only a single strand instead of the nice double helix here. The main primary role in biology of RNA is to translate into the language of proteins, which are actually made of amino acids. And you can see in this graphic here, it's very different. Proteins are highly functional. There are 20 different units of proteins called amino acids. So these big 3D shapes that are translated piece by piece from this strand into a corresponding protein, they're very diverse, and they're basically what accomplish everything in our cells. So whenever you see a gene, you hear the word gene, the basic definition is that it is going to encompass all three of these molecules, and then the entire gene is this process. So say that you have a gene in this section of DNA, it's going to make this RNA right here, and it's going to translate to this protein. So the key to making mistakes that are going to drive growth, let's say that you have one of those stop and go signals, sometimes all you need is to make a mistake, like this, right? That mistake in the DNA here is going to translate to a mistake in the mRNA. That's going to translate to a mistake in the protein, okay? What that means is that if you have a mistake in this functional protein that actually accomplishes, biochemical jobs in your cell, things are going to start going wrong. If this mistake is in one of your stop or go signals, you start to ruin that balance that, controls one cell becoming two, and just that, and it stops right there. So let's take two example looks at genes that can throw that balance off. One is a pretty popular growth gene. So it's an oncogene. And remember, oncogenes are normal genes in your cells. These don't come in, these aren't viruses or anything. They don't come in and change anything. EGFR is a growth gene that is very necessary for your cells. And so under normal circumstances, EGFR is going to promote cell proliferation, promote cell survival. This is very important for a lot of cases, especially, let's say, like wound healing, for example. You need more cells to show up. However, when you get a mutation that locks this gene in kind of a locked conformation, it stays on permanently. That means you're having these effects permanently in any cell that has a DNA mutation that is causing this supergrowth gene to kind of form. And remember, this is going to happen at this level, but it will translate down to this. So all these little cartoons here, these are proteins, and these are going to get affected by the protein that has a mistake in its DNA. Okay. So for another example, P53. P53 is a gene that is going to detect damage, and it is going to, for example, right here, stop the cell cycle. That's a very important role of P53. If you have DNA damage in P53, you will lose this function, all these functions that you see here, and the cell will be able to run wild. If you lose the ability to stop the cell cycle and say, hold up a minute, we need to stop, we can't have these cells growing because this has incurred a DNA mutation that causes this full protein to just, lose itself, this cell cycle is now basically uninhibited. It's unchained at this point, and it can go completely on its own. It has it, the stop and the go signal balance is completely gone. Okay. Now progressively, tumors will pick up, single mutations. Eventually, one of those single mutation cells is going to get a second mutation. These have a growth advantage all of a sudden, perhaps. And then in that population, you get a third, and then you start comprising the tumor. It's usually about 3 to 5 mutations that it takes to show with a symptomatic tumor in a patient. At this stage, these cells are their own population. They have completely broken away from the body at this point. They have their own genetic drivers, and they are completely unchecked when it comes to proliferation and survival. So this first figure up here does a really good job of showing that increasing amounts of DNA changes are going to lead to a more aggressive cancer. What this figure does here really well is shows that you start with a single cell that has a mistake in it, right? And this single cell is going to split, and some of its progeny are going to go off and make different kinds of tumors. So you can see here it is kind of a phylogeny where there are failed tumors in a lot of cases, but some of them do reach the point what we call metastasis, which is where the tumor is so powerful it starts to go into other regions of the body and interfere. That's the end stage for a lot of patients, and this is what we want to avoid. What we know now is that the genetic changes that are codified in this tumor, for example, let's say we defeat this tumor, which we do a lot of the time. We do get rid of a lot of metastatic tumors. That doesn't mean that we defeated this clone right here. And that doesn't mean that it can grow now into this one instead, right? And then you have a resistant or, a refractory tumor that we don't have a therapy for, let's say. So the power of genetic evolution is on full display in tumors. You are fighting multiple populations at once when you are fighting a tumor, because some are more genetically evolved, some are less. And it's actually the ones that are less genetically evolved that are so dangerous, because they can always become something new at that point. And that's what's tough. Okay, this is a good comprehensive kind of final look before we get into the specifics of what my work is, about what the hallmarks of cancer are. The main ones that I kind of showed you, you need to be able to resist cell death, because like I said, there are a lot of signals that just, if they can tell a cell is having a hard time, the cell will just explode. And then you also need to be growing and multiplying at a very high rate. The rest of these are very, very interesting, and trust me, I could talk about every one of these and why it's so interesting. And so could any other lab out in the world that is working on this disease and these diseases. But as you can tell, that would take up too much of your time today. So we're gonna focus mainly on the genetics that I cover in lymphomas that make that make us able to predict patient outcomes. Okay, this is a survival curve for the disease that I study, that I usually use the acronym of DLBCL. So diffuse large B-cell lymphoma. This is the most common lymphoma there is. There's about 35,000 cases a year. About 50% of those will actually get cured. And I don't use that word lightly either. It's very good, right? There are two variations of this disease. GCB in blue is the less aggressive variant. Anything non-GCB, this is an aggressive version of this disease. This presents clinically and genetically. So if we break down the survival plot right here, this Kaplan-Meier here, let's take a look at a 2-year mark for progression-free survival. With the aggressive version of the disease, about 30% of patients, so here's about 30% right here, they are still going. With the less aggressive version, you have, close to 70% still going. So that's great. So we've already kind of genomically classified this disease. The key right here is helping these patients, though, since they face such a dismal outlook just with the standard therapy that we're going to give them. And we'll introduce that therapy in a sec, too. But so for I know that this isn't a true biology presentation, but what DLBCL is that your immune cells are in a constant cycle of growth and survival as they are priming and educating themselves against new pathogens. When your body encounters a pathogen, it starts B-cell development. And that B-cell development can get out of control. And that's where you get something like a DLBCL here, that you have cells that can survive and they grow way too fast. And there are a lot of processes in that biology that I'd love to explain in an immunology video. But for our purposes, we can keep taking a look at just at the pure genetics. Okay, so like I said, about 50, oh, that cut off, about 50% of people are going to make it by just using a therapy we call R-CHOP. R-CHOP stands for rituximab, which is an immunomodulatory CD20 antibody. So this is going to target your immune system to kill any cell that has a CD20 molecule on it. That is a marker for B cells. So that's great. That's going to kill off a lot of the bad B cells. Perfect. CHOP is an acronym for 4 different chemotherapies. They are very good at getting rid of, cells that are aggressively replicating, and one of the things they replicate is their DNA. So one of the jobs of these 4 is to kill cells that are over-replicating DNA, and that's the basic job of any chemo. So using this regimen, you are curing about 50% of these patients. Now the problem is that if you do not receive a cure from R-CHOP, this is your outlook. This was a paper that was in 2017, and it kind of woke the field up and said, okay, like we have made very little progress when it comes to anyone that doesn't do well on R-CHOP. This is where we're at. At a 2-year mark, we only have about 20% of patients that haven't had another event. This entire drop-off is incredibly steep. We lose 80% of our patients in those 2 years, and that's unacceptable from a medical standpoint. What we need is not only therapies that can address these patients directly, but we need to make sure they never join this population. So what that means is that we need better frontline therapies than just R-CHOP. Now R-CHOP's great, but we need to modulate it. We need something better. We need a targeted approach. So let's say you have a patient here. What we need to do is screen their genomics. We need to know exactly what DNA mutations they have, what genes in the RNA are actually very active, and then we'll get to that tool later. We need to add a targeted agent to this R-CHOP backbone. So we know R-CHOP works. We don't want to just change the therapy. Add something to the backbone depending on the genetic results, and that is going to tell us and inform us how to best target. So in this case, imagine this little schematic is going to show us we have a patient, we sequence them, and at this point we can make a decision. Is their sequencing in line with, okay, this is going to respond to regular R-CHOP, or are we going to need to add a different therapy to this? And that's what a targeted agent is. It's another drug. It's another medicine, basically. So in the last couple of years, there have been a big number of big integrative analyses of DLBCL. This is an example, Chapuis 2018, where they took about 304 cases and genetically classified them. That allowed us to predict outcome. We learned a lot about the biology based on what DNA mutations were driving each of these 5 clusters right here. And we also, this is the big one, we started to say, okay, based on, let's say, this C3 right here, which you can see right here, we should add a targeted combination therapy. In this case, we're going to inhibit the oncogene PI3K alpha and the oncogene BCL2 because those are enriched in this C3 column right here, these patients right here. This is the beginning of a targeted therapy idea, is that we have the genomics now to do this. We just have to apply it. So what we did is we gathered patients from the Mayo Clinic. Now each of these patients have been sequenced for their DNA and for a lot of them their RNA as well. We have 149 patients treated with standard R-CHOP. Oops. We have 47 patients treated with something new, R2-CHOP. The 2 stands for what this drug is right here, lenalidomide. Because its drug name is called Revlimid. So rituximab plus Revlimid, R2-CHOP. What R2-CHOP is a new targeted agent added on top of the backbone of the existing R-CHOP regimen. What lenalidomide is, it is an immune modulator. This excites the immune system and it kills aggressive cancer cells. But we needed to see if this was going to work in a specific populations of patients, right? So we have 2 factors that we divided our study based on. EFS 24 is if a patient successfully made it without a relapse event, so the tumor coming back, for example, in 24 months, so 2 years. If they failed, that means they have an event in those years. Well, whoops. But if they achieved it, that means they did well. They had a good outcome, right? Oops, let's get rid of that. Okay. And then our other factor is which therapy we gave them, and we have those here. We have R-CHOP and we have R2-CHOP. And this is the key question. What DNA mutations are causing people to fail R-CHOP but are found in people that are doing really well with R2-CHOP, right? Because that's a key question. We need to know who would normally fail standard therapy, but who would extremely benefit from this new therapy. And you can't just give lenalidomide to everybody. This is a pretty tough drug. This causes some side effects. You don't want to just hand this out. So, for any clinicians in the room, I did figure I should show you the nice clinical tables. You can see this in the publication. Super fun. I'm not going to sit here during this presentation and look at it. You can pause if you want and learn a little bit about the clinical population. For the most part, it's a nice balanced population, but it's basically the stats that we were looking at just a sec ago. So we took all of these patients and we had their DNA and we sequenced a targeted panel of 212 genes that are highly associated with lymphoma. These were basically annotated from 3 major publications that sequenced the entire genome, and each identified about 100 genes that were, these are drivers of lymphoma. These are things that are driving the pathways that lead to growth and proliferation in lymphoma. So we decided to target those. They had already done the big work for us, so why ignore their data and go in? So we took a look, and we cross-compared those 2 factors that we talked about. On the x-axis, you have the association of each of these little bubbles is a type of mutation in a single gene. On the negative, on the left here, so here's your axis right here. That means that all these mutations on the left side of this axis, they are associated with a poor EFS24 response. That is a bad response, okay? So things like mutations in the CD79B or BRC6, those are bad mutations if you're taking R-CHOP. On the other hand, we also took a look at R2-CHOP association with EFS24. Anything below this axis was associated with a poor R2-CHOP. So the addition of lenalidomide, these patients did not have a good response when lenalidomide was added. And they also had, this tumor was in, or this gene was mutated in their tumor. That means that we have 4 sets of data to look at. So let's start over here in the red. These mutations are in the negative for both. If you have one of these mutations, things are not looking good regardless of the therapy we are going to give you. Like we highlighted, TP53, remember how that was such an important gene to keep tumors in control? It didn't matter if you were treated with R-CHOP or R2-CHOP. If you have a TP53, we're in trouble and we need a more aggressive therapy probably. So up here in green though, all these mutations on this side of the positive axis, if you had one of these, it actually meant that your tumor was pretty responsive, in the growth pathways that it has, pretty responsive to either therapy. So that was good. So we identified those genes. Over here was a strange set. These are genes that are associated with a very good response to just the standard therapy, but not so much the R2-CHOP. So this is actually very good data to find because, like I said, you don't want to give R2-CHOP to everybody. Let's say you have a mutation in BCL2, an oncogene. Perhaps you don't want to give the R2-CHOP in that case. Now, there are ways to score that, and we'll take a look, but let's take a look at the main focus of this paper and my work. These are mutations associated with failed R-CHOP. So these are patients that would join that really terrible curve that we showed you, right? But not with R2-CHOP. With R2-CHOP, they're high in the benefit. See how this scale, see how the y-axis is all, if you go up this direction, that's a very positive thing for R2-CHOP. But like in R-CHOP, they are far over here. They're way over here. So what we found is that based on a statistical enrichment, these 3 genes right here were associated with a terrible response to the standard therapy, but a great response to the new therapy. So each of these has a different role in driving the tumor. MYD88 is a classic driver of lymphomas, very aggressive ones. It is an internal signaling molecule inside the cell, so it's very hard, if not impossible, to drug. Most of the drugs that we do affect things that are on the surface of the cell. PIM1 is also something that gets mutated pretty often, and there are different pathways that it modulates or is suspected to modulate. But for the most part, we know that it's associated with some pretty aggressive tumors, so we figure the role is somewhere in that line. Now, SPEN is one that I know this presentation won't be able to focus on, but SPEN was something new. SPEN isn't something that people know a lot about when it comes to lymphoma, but here it is, highly associated with a terrible R-CHOP response, but a great R2 CHOP response. SPEN, what we know, what I can tell you from him now, is that it is associated with the Notch signaling pathway, which can drive growth unintentionally once the tumor gets to a certain advanced stage, let's call it, in these terms. So let's go down here. So if you have one of these mutations, take a look on the right. If you have one of these, what I call reversal alterations, that's why I call them RA, they're in red. If you have standard therapy and you had a MYD88, a PIM1, or a SPEN, that is your outlook. And at 24 months, we have about 30% of our population left. That's it. So this is a terrible outlook, highly significant difference between these populations surviving. So if you have one of those 3 mutations, things are not looking good. But if you transition over to here and you look at the red, every one of those patients, all 8, they all made it. And that's where it gets important to define these differential responses based on genetics. Because we can find areas of clinical, benefit, basically, not just for, you can't just treat everybody with the same thing. You have to identify what's going to work based on genetics. And that's what we did here. And that's why the power of these 2 curves right here is so, immense in my mind, is that this is a very good example of saying, okay, we can take these patients and we can put them over here if we just treat like this. We can save a lot of lives and a lot of time, all kinds of stuff, if we, start personalizing medicine and we take this to the next level. And we are, because like I said, we have new tools that we can do this with. So in this case, yeah, like you see, the red curve is doing so much better than the red curve over here. So we took a look at DNA mutations. Remember that DNA codes into that RNA. We can measure that RNA to see what is high and what's low, for as far as genes. So remember, genes aren't just on and off. They can be on a lot, they can be on a little. So in this case, we did something very similar. We have an R2 CHOP EFS24 association and an R2 CHOP association. I think I said, yeah, R-CHOP right here. That means that, again, these genes right here that are on really high, when these are on really high for patients, that is a bad signal. If you are taking just the standard R-CHOP, oh no. But if you are taking the new regimen with lenalidomide added, these actually become beneficial markers. So you can completely change the game on the tumor as long as you know that some of these marker genes are driving, your lymphoma essentially. And so using data like this and like this, we can start to predict not only who's going to respond, but we can actually see the biology of the tumor as well. We can see who's responding against a modulatory drug and a non-modulatory. So in this case, we have associations with different tumor driving pathways, because there's a lot. And in R-CHOP, we're in red. So if you go up, that means there's a positive association. That means you're doing well. You're reaching that EFS24. So for example, here's MYD88 upregulated. So not only are we looking at MYD88 just if it's mutated, but if it's really high and the genes that it's driving are up high, if that's the case, and you have R2 CHOP, you are looking at a great outlook, because the higher this association is, the higher it is associated with patients that did reach 24 months. You can see here with just R-CHOP, there's kind of a margin of error here that it's like, we're maybe not doing so great. What we can also see is where immunomodulatory things like lenalidomide are not working as well. And we can see that in some cases where you have like a microenvironment with lots of stromal cells here, or you have genes that aren't responding to lenalidomide that are really high, that's a chance again where you don't want to use R2 CHOP. So on one hand, R2 CHOP is amazing if you target it to the right spots, because you don't want to target it to the wrong spots either. So that's the point of individualized medicine, is that you also are going to know which patients not to give certain things to. So here we are. This is the basic idea and the pathway that we identified, is that there are some big big master players at the end in the nucleus. They turn on a lot of genes, and they are going to, they're going to play a lot of cards. They're going to be the ones that are pulling a lot of strings. We found in green here, these are our reversal alterations that usually are driving a bad phenotype unless we give them lenalidomide. So again, here, these are kind of the proposed roles for each. We know a lot about MYD88's role. And with lenalidomide, we're basically seeing that genes that drive growth like CART11 or JAK, for example, JAK-STAT signaling, we can inhibit those with lenalidomide where we couldn't before. But we need to identify that pathway is being activated, that these pathways that we saw, by the mutations in these genes. So only then can we really, validate that this is a good application of the R2 chop. Okay, and this is kind of back to where we started. Now in a future of tumor medicine, we can take a patient and we can genetically screen them right here at the beginning, right? We can see, let's say we go over here and we detect, oh, I don't like when that happens. There we go, sorry. Oh no, I was so close to this being okay. All right, so we can detect, okay, they have one of these mutations, for example. They have this inflammatory profile, which was what I should have called that RNA profile we were kind of covering. Let's put them on R2 chop, okay, perfect. Now, part of the study that also uncovered, hey, we found a lot of R-CHOP responsive genes. That's perfect. If we screen this patient, they don't have these, they have a pretty standard R-CHOP responsive genetics, hey, let's do R-CHOP. And remember, in red, like we saw, there are genes that are predictive of failure of both of these therapies. That means we have to probably immediately turn to something more aggressive because we don't want to waste our time, and put the patient through things like these when we could probably save time by turning to something more aggressive. So at this point is the key right here, is just saying we can take genetics and use them to predict response. We can use them to predict what a patient is going to do best on and respond to. And this is an exciting future of, not only cancer medicine, but just about anything, right? If you can apply genomics at a patient scale for any application, you can improve response, you can improve, whatever you're looking for based on every patient. And it's kind of it's like the full extension of the Hippocratic Oath, that every patient is different, right? And that's what, makes me so excited about this. And, I'm glad that you joined me on a fairly complicated and, kind of pretty clinical look into things. But I really enjoyed it. Thank you so much. Please feel free to email me at my Gustavus email for any of the questions, discussions. I could do like a follow-up video or kind of post something in the faculty L or something, and would love to hear from you. So thank you so much for watching, and I hope your quarantine is going well.